I want to begin by looking at a view of life. In this chapter, we will define life, describe the emergent properties of living things, looking at how they gather materials and energy, how they reproduce and develop, how living things can adapt and go through the process of natural selection. We'll also look at the organization of the biosphere and how we name scientific organisms, the taxonomy, and then take a brief review of the scientific method. So if we start out with a definition of life, every living thing is composed of the same chemical elements. This would be hydrogen, oxygen, carbon. They also must obey the same laws, both physical and chemical, as any non-living. If you put a ball down on the ground and kick it, that ball will continue to roll until a force stops it. Usually friction is what stops it. That is a non-living object obeying one of our physical laws. Well, living things obey the same law. Objects in motion stay in motion. That's an example of how living things have to obey the same laws as a non-living thing. So looking at the characteristics, we know that very first thing, all living things are organized. You begin with an atom. Atoms can make up molecules, a molecule of oxygen, a molecule of hydrogen. You put together those molecules and you build a cell. Now a cell is what we consider the smallest, most basic unit of something that is living. Even non-living things are made of atoms and molecules, but only living things are composed also of a cell. Now some of our living things are unicellular, whereas uni refers to one cell, while others are multicellular or composed of many cells. Humans are an example of a multicellular living organism. Now if we take cells and put cells together and they function for a common cause, they make a tissue like epithelial tissue, muscle tissue. You put those together and they make organs like the skin, the heart. You put multiple organs together and you make an organ system. What we're looking at is referred to as emergent properties. Qualities that a living thing has. These qualities may only appear or will only appear as each level of organization becomes a little more complex. Each level has properties because of the way the different parts are interacting. For example, a cell has certain qualities. When you put cells together to make tissues, tissues have more complex qualities that are only seen because we're putting together cells, parts of the whole to make a tissue. These emergent properties explain the complexity of living organisms. So we know that living things are organized. Our second characteristic is that living things can not only acquire or get their own food, but they can then process that food. Now, why must living things get and process food? Because living things require energy, the capacity to do work. And that's not necessarily physical work, like you're thinking about getting up and doing work, but even your cells undergoing basic metabolism is considered work. Now, metabolism is any chemical reaction that occurs in the cell. That chemical reaction could be the building up of something or the breaking down of something. Metabolism just refers to change. We need this energy. We need to undergo metabolism in order to maintain the functionality of a cell. Now, the ultimate source of all energy for us is the sun. The sun is very important in the process of photosynthesis, where we're going to transform solar energy into a chemical energy. And then we living things take in that chemical energy and use it for metabolism. Our third characteristic, living things respond. Now, in particular, they respond to some kind of stimuli. Living things not only respond, but they have the ability to maintain these conditions within boundaries. And we refer to this as homeostasis. So think about this as being in the same place. This is actually place, sorry. Homeo, same. Think about all the things that our body maintains the same approximate place: heart rate, respiratory rate, glucose level, sugar level, blood pressure. That's the ability of our body to maintain homeostasis. Now, living things have the ability to detect changes. The human body is a little easier to understand because when we go outside and we get hot, that is a stimulus that our body detects, and our body then begins to maintain our normal boundaries by allowing us to sweat. The sweating will cool our body and help us maintain homeostasis. But your textbook also refers to a lot of other examples of responding to changes in our environment. Probably the easiest for you to understand would be a plant following the sunlight. You can take a plant and place it near a sunny window, and if you never turn the plant, you will look at it over a course of a few months and notice the plant has actually bent or is leaning in the direction of the sunlight from the window. That is an example of responding to stimuli. And we refer to these responses as a behavior of a living thing. So we know that living things are organized. Living things can acquire materials and energy. Living things respond. Our fourth characteristic is that living things reproduce and they also develop. Now, reproduction is necessary in order to continue a species. So it is a major characteristic of living things. We know that a lot of multicellular organisms have to begin by uniting two gametes. And your gametes, for a human, would be your egg and your sperm. We also know that we take these from two different individuals. It allows us a lot of variation or variety in the living population. Now, development is controlled by your genes, these instructions encoded within our genes that tell the body how to develop, when to develop, how far to go. And your genes are just portions of DNA, these really long spiral molecules found inside of a cell in a region referred to as a chromosome. We also know that living things adapt. Any modification, any change that makes an organism better suited to live in a particular area, a particular environment, is referred to as an adaptation. Now, over time, those organisms may become modified because nature has selected that particular characteristic to allow them to live in that environment. One of the best examples would be sickle cell anemia. Now, in America, sickle cell anemia is detrimental. It's harmful to your health. It's a disorder of the red blood cells that causes them to change their shape. They don't carry as much oxygen. They tend to get stuck in your little tiny capillaries, causing chronic pain. It's not a benefit. But sickle cell anemia at one time was an adaptation that allowed people to survive malaria-ridden areas. If you lived in a malaria-ridden area and had sickle cell, you did not contract malaria. So the so-called healthy people without sickle cell ended up dying from malaria, whereas those with the sickle cell survived. This is an example of natural selection. Now, over time, that change may become prevalent throughout the entire population. If the entire population takes on this adaptation, we refer to it as evolution, a change over time. Sickle cell anemia is not considered evolution because the entire human population does not now carry sickle cell anemia as a defensive mechanism. So living things are organized. They can acquire their own food and process that food to get energy. Living things can respond to stimuli. They can reproduce and they can develop, and they have adaptations to make them better suited. So how do we classify our living things? Well, the whole science of classifying organisms and identifying them is referred to as taxonomy. Now, we have levels of taxonomy that are based on evolutionary relationships between species. Now, if you start out with the least inclusive, meaning the most specific, then we have something referred to as a species. So we want to go to the other end. Let's look at the broadest category, the biggest category, which would be domain. This is the largest, the most inclusive. Think about this as really, really broad. And think about this one as really, really, really specific. For example, the domain eukarya is the domain we humans belong to. So do mushrooms, so do mice, cats, dogs, plants. You know that we are very different than a plant, but yet we are all in the same domain. It's a very broad category. The next category then be a kingdom, kingdom Animalia. We are animals, mice are animals, dogs are animals. Kingdom Plantae, that separates out and puts the plants in a little different category. If we go a little more specific, we have a phylum, down to a class, then an order, then a family, a genus, and lastly a species. Now there is a little mnemonic device, a little rhyme to help you remember this order. If you can remember that Dear King Philip, the mnemonic device just uses the first letter to remind you of something, came over for good spaghetti. If you can remember the little rhyme that Dear King Philip came over for good spaghetti, then you can remember that we have a domain, a kingdom, a phylum, a class, an order, a family, a genus, and in the most detailed, a species. Every living organism is categorized using this taxa, these levels. So let's look at the domains in a little more detail. We have three main domains. We have the domain bacteria. Now, this domain includes tiny, tiny, tiny unicellular prokaryotes. These microscopic one-celled organisms were developed before we saw a nucleus appearing in cells. So when you're referring to a bacteria, you're talking about something so tiny we can't see it. If so, we would never get sick. They're composed of a single cell, and they're a prokaryote. They existed before nuclei. They also reproduce asexually, meaning you can have a bacteria on your counter, do nothing but provide it the right conditions, come back in 10 minutes, and where there was one, there's now 100. It does not need a secondary organism for reproduction. The next domain is archaea. Archaea are also unicellular prokaryotes. They're a lot like bacteria. They also reproduce asexually. But where they're different is they're found in very extreme aquatic environments. We have the methanogens. They find areas that are rich in methane gas, and that is where they live. The halophils. Phil means to like or to love. Halo refers to salt. You find a halophil in a very salt-rich area, an area where you would not have anything else living. Whereas a methanogen might be found in a swamp or a marsh, you would find a halophil in the Dead Sea. Then we have our thermoacidophils, to like or love. This is referring to acid. And thermo is heat. So we find these in heat, acidic, rich areas, geysers, volcanoes, places where you would not find any other living organism. Our last domain is the domain eukarya. Now here is that term karyo again, nucleus. And eu actually means true. So we see the appearance of a true nucleus in the cell when you reach the domain eukarya. They can both be unicellular or multicellular. But the key characteristic is that their nucleus is bound by a membrane. Typically, you will see sexual reproduction in the domain eukarya, and we have our four kingdoms. We have our Animalia, our Plantae, the fungi, and the protist or the protista. For bacteria and archaea, we're still not sure about the kingdoms. So if we're not sure about the kingdoms, we can't classify any further. If we don't know a kingdom, we don't know a phylum, a class, an order, and so on. When we classify bacteria, They're simply classified the domain bacteria. The same thing for archaea. But eukaryotes can be described even further because we have the protista, the protist, we have the fungi, your mushrooms, your plants, your animals. So once we continue to go down our taxa, our levels, from our domain to our kingdom, to our phylum, class, order, we're going to eventually get down to where we can assign a living thing a two-part name. This is referred to as the binomial nomenclature. Nomial referring to a name, bi meaning to give it two. I'm going to give it two parts to its name. And this scientific name consists of our most specific descriptors, our genus, and then our species, which is referred to as the specific epithet. When you're looking at a scientific name, the genus and the species, there are certain rules when you're writing your scientific name. First of all, the scientific name, if being typed, must be in italics, where the genus is capitalized and the species is lowercase. If you are hand writing this, you follow the same rule, genus, space, species in lowercase. But because we cannot write in italics and cursive does not count, we must underline our genus and our species. For example, the garden pea. The garden pea, Pisum sativum, would properly be written like this. Because I am hand writing, I must capitalize the genus, make the species lowercase, leave a space between them, and underline the two. Now, science chose these odd sounding names, Homo sapiens, Pisum sativum, which is a Latin-based language. Now, a couple of reasons why we've chosen Latin. First of all, it is universal. It allows us to avoid confusion. If you say, I'm going to see a football game, then you may think football completely different than the next person. I think football, oblong, pigskin, pads, helmet, quarterback, offense, defense. But if you are overseas and you go to see football, you're looking at what we call soccer. Anytime the language is not universal, there is a chance for confusion. So by using a dead language, which is what Latin is, means you're using a language that does not change. There's no variation. If you would have said 5 years ago, tweet, we would have all thought of a bird, tweet, tweet. Well, now tweet means something completely different in the technology age. So we want to use a language that's not evolving, that's not changing. So Latin is the choice of science. When you're writing this scientific name, you can give just the genus, because it gives you a big broad group. But you cannot give just the epithet. You cannot simply give the epithet alone. Because giving just the epithet is like giving someone a house number without giving them a street name. It's like saying, come pick me up at 200, I'll be waiting on you outside. Well, think about how many 200 address numbers there are just in this region. By giving them a street name and a number, you're giving them a very detailed description. Now, we said we could give the genus alone. Well, say, pick me up on Jones Drive in Dothan. You've given them just the genus, the street name, but yet they can ride down Jones Street and pick you out. We can give a genus alone and narrow down, but never give just a specific epithet. That'd be like giving them just a number without a street address. Your first lab assignment is going to work on using this binomial nomenclature. So make sure you understand your basic rules. Genus, capitalized, space, species, lowercase, underline both. If you're handwriting, if you're typing, it is acceptable to put them in italics. So let's flip now and look at the organization of our biosphere. Because we've taken a cell and we've made it into tissues, and our tissues have built upon and made organs and organ systems, all the way until the point we get to a organism, a member of a species. Well, organisms are considered all members of a species that are living in a particular area. In a particular area, all of those organisms are called a population. So if you refer to the population of Dothan as, say, 60,000, you're talking about all members of a species. We're talking about the human species. The human species of Dothan would be 60,000. That would be our population. Now, there are several populations in Dothan. We have populations of trees. We have populations of cats, dogs, mice, roaches, bacteria, fungi, a lot of different populations. If we consider how all of these populations interact within the same environment, then that is a community. So when you're talking about the community of Dothan, you're referring to all the species living within that particular environment. The dogs, the cats, the roaches, the mice, the fungi, and the humans make up the community of Dothan. Now, there are a lot of different communities. We have the community of Dothan. We have the community of Wicksburg, Quincy, Headland, Ashford, Slocum. All of these communities within the same physical environment can make up an ecosystem. Now, we could not use our example and use, say, Huntsville, because Huntsville is not in the same physical environment we are. They're a different altitude. They're a different latitude. They're a little further north. They get more cold weather. That does not make up that ecosystem. We in this area of South Alabama, we refer to as the wiregrass, would be an example of an ecosystem. In the Huntsville region would be another ecosystem. Now, if we take all the ecosystems of the earth, then we have what's referred to as the biosphere. In our biosphere, we're going to see a lot of biodiversity. Now, we have about 15 million species around. The total number of species, their variability, how they're different, and the ecosystems in which they live make up a very complex biodiversity. Now, that's necessary. We are using this biodiversity to ensure our survivability. Because there's a variance in humans, we can survive different things. Think of someone who is very sickly with a very weak immune system. If the entire population didn't have a variability, some strong, some weak, then something like the flu could wipe out the entire human population. That would lead to something referred to as extinction. Now, we do not have as much extinction because we have a lot of biodiversity. A lot of people say that preserving our ecosystems is one of our primary bioethical issues right now. We do modify the ecosystems for our own purposes. We clear the land to build homes. We clear land to build malls. We use the wood to build our actual structures. We need to be careful about maintaining our ecosystems to ensure that we have a lot of biodiversity. Then we want to look at our scientific method. We want to go back and kind of quickly review the scientific method. It should have been covered previously, most likely in junior high or high school, but we want to take a broad look at the scientific method. The scientific method, a way we scientists go about solving a problem, always begins with some type of observation. We see some kind of phenomenon, some natural event that we want to understand more. Now, we can use our senses, our five senses, sight, touch, taste. We can even use instruments like telescopes to make our observations, microscopes to make our observations. But once we've made an observation, we want to try to explain what we are seeing. So a scientist to explain an observation or a phenomenon would create a hypothesis. Now, we've heard of hypothesis before, educated guess. Well, here's the thing about a hypothesis. We want to use what's referred to as inductive reasoning, meaning we want to use creative thinking to combine a lot of isolated facts into a more cohesive picture. You want to be able to take this idea and that idea and that idea and put them together and say, if this, that, and that work together, then I expect this to happen. Now, we do present it as a statement. We do sometimes say, if, state what we think, then state our expectations. Now, the things about creating a hypothesis, they have to be testable. You have to be able to create an experiment to test that. If you notice that the lady next door has hummingbirds everywhere, and you think, okay, she has hummingbirds, I'd like to have hummingbirds. She has a garden full of beautiful flowers. She has hummingbird feeders everywhere. You start to make an observation. There's a phenomenon. She has hummingbirds, you do not. And you want to create that same environment. Then you would put together all of these isolated facts. She has flowers, she has hummingbird feeders, and try to make a cohesive whole out of it and create a hypothesis. If I place hummingbird feeders in my yard, then I too will have hummingbirds. That's testable. We can place hummingbird feeders, we can test that. If that doesn't work, we could try something else. If I plant flowers in my garden, then I will have hummingbirds. Now, notice you cannot test moral or religious beliefs. You can't test opinion. If you look at a piece of art and say, that painting is beautiful, you cannot make a hypothesis and you cannot test that, because that is opinion. Your opinion of that painting and my opinion of that painting could be two different things. We also can't test religious beliefs. Religious beliefs cannot be turned into a hypothesis and then experimented on. So we'll take our hummingbird experiment, our hummingbird hypothesis, and we need to create an experiment based on that. So we want to create a design. Now, every experiment in science has to follow certain rules. We want to follow rules to make sure that, one, our results have meaning, and two, that they're repeatable. Because for science to accept your study, other scientists must be able to repeat the work and get the same results you did. So when we create an experiment, we're going to divide our subjects into two groups. We're going to have a control group, that way we know our experiment's working, and we're going to have an experimental group. Now, the control group is going to have everything stay the same. The experimental group will have everything stay the same except one variable. Now, you can only test in science one thing at a time, because if you test multiple things, you're not sure really what created your results. You can't, or you shouldn't, in our hummingbird example, go out and plant a garden full of flowers and put out 5 different hummingbird feeders and add 3 bird baths to your yard. Because you would not be sure if the flowers or the bird baths or the feeders brought the birds to your yard. So therefore we want to test one variable, change one thing at a time. When we do this, it's referred to as a controlled study. And a controlled study has 2 variables. All right, the experimental tells you what it is. It's the one we experiment with. This is the one thing we are changing. So let's say we want to run an experiment with the hummingbird feeders. Well, science says I have to have a control group. So what do you really think about the hummingbird feeders attracts the hummingbirds? Is it the feeder, or is it that sugar water in the feeder? So we're going to have to do research before we do our experiment. And we're going to learn that hummingbirds like the sweet nectar. So to run a proper experiment, our control would be a feeder with plain water. That's normal. That's the same thing a bird could expect in a normal condition. We didn't change anything. Water falls from the sky, plain water. In our experimental group, we're going to take our hummingbird feeder of water and we're going to add 1/4 cup of sugar to it. The plain feeder is our control group. The feeder with the sugar in it is our experimental group. So what actually is our experimental variable then? What is the factor that we're testing in that setup? Is it the water? Can't be the water, because the water is the same in both groups. We're actually testing the sugar. The sugar in the water is what we think is attracting the hummingbirds. So we set up our 2 feeders, we run our experiment, we count the number of birds that come every day, how much fluid they drink, etc. And then we come up with some result. The result is referred to as our response variable. Think about this as your result. Now, it's also referred to as the dependent variable. So let's consider why dependent. Think that dependent means it depends on something. So the number of hummingbirds, the result, is going to depend on what? It's going to depend on the presence of sugar. We would expect more hummingbirds to the feeder with the sugar water. The number of hummingbirds to the sugar water is our dependent variable. It's also called our response. It's the response we saw when we added sugar to the water. So we're going to collect data. We're going to count the number of birds, how much fluid they drink, same time every day, same amount. Now, when you're setting up that experiment, in order to make sure your data is viable, you need to make sure that you didn't change anything but one factor. Your water has to be the same temperature. Your feeder has to be the same color. Your water has to be from the same source. You can't use distilled water in one and tap water in the other. You can't stir the sugar one day with a spoon and the next day with your finger. All of that is adding new variables. So if we know that we've added only one variable, the sugar, everything else is constant, then we can collect data, objective results. Now, if you notice a lot of variance in your data, a lot of variety, there's probably an error somewhere. So we need to take a look back over our results. Now, typically we display our data on a graph or even a table. Then we analyze our data and we come up with a conclusion. Now, we're going to interpret our study and figure out if our hypothesis was correct or not. Well, if our plain water attracted 2 birds and our sugar water attracted 200, then we can conclude that if I place sugar water in my garden, then hummingbirds would come to the yard. We have a valid scientific study then. Now, typically we would go on and publish our results in a scientific journal. It would be open for inspection by other scientists. It should be repeated by other scientists for validity. That leads to what we refer to as peer review. You want your peers to review your work. And by reviewing your work, that ensures that you not only did your steps correctly, but that the scientific community will accept your data. So once we do these, we're really out for that ultimate goal of science. We're just trying to understand the natural world. And we're trying to understand it and explain it in scientific theories. Now, what is a theory? Because we really use the word theory improper. Because you may go out and go to crank your car and it not crank, and you say, I have a theory about why that's not working. Or, I have a theory about why he doesn't like her. Well, that's not really what a theory is. A theory is not your guess about something. A theory is actually 2 or more hypotheses, explanations of something that are related. Now remember, a hypothesis has to be tested. It has to be proven. Data was collected. A conclusion was drawn. That's why our old use of theory is such a bad term. A theory is taking 2 or more hypotheses that are related, that have observations and data to support them. We have the theory of evolution. We have the theory of relativity. Those are more than one related hypotheses that have been proven true, that do have data to support them, that are connected together. Now, if you can get a theory, or even a group of theories, that the overwhelming majority of the scientific population agrees upon, then we call it a scientific principle or a law. A law is a theory that everyone agrees on. That is why the theory of evolution will most likely always remain a theory. You will never get the overwhelming majority of the scientific population to agree with the theory. Therefore, it will never become a principle or a law. We have the law of inertia, the law of gravity. Now keep in mind that scientific laws can change if We come up with new experiments, new hypotheses, and so on. Now, do I expect the law of gravity to change? Not in my lifetime. But keep in mind that scientific laws can be modified if necessary. So let's review this chapter. We defined life and talked about the emergent properties, the properties that only appear as each level becomes more complex, more complex, more complex. We looked at the characteristics a living thing should exhibit. They should be organized. They should be able to acquire their own materials, get their food, create energy. They should reproduce and develop. They should be able to adapt, and natural selection will play a role in that. We looked at the organization of the biosphere, how we have communities that make up ecosystems and so on, and how that biodiversity leads to a greater variability in the human species or any living species, which increases our chances for survival. We looked at classifying organisms by a domain, a kingdom, all the way down